أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. We are discussing a little bit about my research work, preparation and optimization of ready to use kit conditions for swab test on animal food. In previous video I have shared about the introduction, and now next I am going to share about the materials and method used in my research work. So a little bit about the methods. First of all, first step regarding my research work was isolation and re-identification of Bacillus subtilis. Actually, Bacillus subtilis was procured from microbiology lab and re-identified through cultural, morphological and biochemical characteristics as described by USDA in 1998. Gram staining was the next method to re-identification of Bacillus subtilis. Gram staining was used. Thin smear of growth was prepared from solid media and heated to fix the smear. Smear was flooded with 1% aqueous solution of crystal violet and dipped in 5% solution of sodium bicarbonate and allowed to stand for 2 to 3 minutes. Flushed off with Lugol's grams iodine solution, covered with a fresh solution of Lugol's iodine, which is left for 2 to 3 minutes and washed in water and did not allow the smear to become dry. Simple method of gram staining was followed. Decolorized with acid alcohol until the decolorizer flowed from the slide almost uncolored. Counterstain using 2% solution of safranin for 10% for 10 seconds that was used, washed with water, blotted and dried quickly. Gram positive purple colored and large rods were observed. Bacillus subtilis, that is gram positive purple color and large rods, they were observed under low power and high power of microscope. So gram staining was used for re-identification of this organism, because this organism is a, has a basic use in my research work. So again, mind it please that gram positive purple color and large rods, they were observed under low power and high power of microscope. Second method for re-identification of Bacillus subtilis used was spore staining method. The pure growth smear was covered with 5% aqueous malachite green and steam for 1 to 3 minutes, avoiding the boiling. Steaming was allowed on hot plate or Bunsen burner for 2 to 3 minutes without allowing the stain to evaporate completely. Washing was done under running tap water. Aqueous safranin 0.5% was used as counterstain for 30 seconds. We are discussing about spore staining method for re-identification of Bacillus subtilis. Water was rinsed and drained. Spore appeared green colored. So spores of Bacillus subtilis, they appeared green colored, while vegetative bacilli appeared red colored. Lipid granules, they remained unstained. Noted please, spore appeared green colored, while vegetative bacilli, they appeared red colored, and lipid granules, they remained unstained during this spore staining method. Next method used for re-identification of above said organism, Bacillus subtilis, used was sugar fermentation tests. For this purpose, mannitol and glucose fermentation tests, they were performed for the re-identification of pure isolates. So mannitol fermentation test, in this procedure, inverted Durham's tube was dipped in sterilized peptone water media with phenol red indicator. Stock solution 5% of the mannitol that was prepared and tenderized, then 0.25 ml of mannitol was dispensed in a test tube. Culture of pure isolate was inoculated during platinum loop, using platinum loop. Culture of pure isolate that was inoculated by using platinum loop, red to yellow color change was observed. Red to yellow color change was observed for acid production, and no gas production was observed, which confirms the Bacillus subtilis. After that, glucose fermentation test that was performed for re-identification purpose. For this purpose, inverted Durham's tube was dipped in 5 ml of sterilized peptone water with phenol red indicator. Stock solution 5% was prepared and tenderized, then 0.25 ml of glucose solution was dispensed in a test tube. Culture of pure isolate was inoculated using platinum loop. Color change was observed from red to yellow. Color change was observed from red to yellow for acid production and absence of gas in the Durham's tube. So this was the procedure adopted for glucose fermentation test for re-identification of Bacillus subtilis. Next procedures were biochemical tests. We are discussing about the research work done by me. Topic of research was preparation and optimization of ready-to-use kit conditions for swab test on animal food, abbreviated as STAF. In this research, 3 things were optimized. Number 1, culture of media that was optimized. Thickness of media that was optimized. Number 2, spore count of Bacillus subtilis that was optimized. And number 3, incubation temperature. These 3 things were optimized during this. But before starting the whole procedure, we have to re-identify the organism Bacillus subtilis, and for that we have to adopt these biochemical tests. Number 1, indole test. For indole test, some organisms utilize the amino acid dissolve 5 grams of p-dimethylaminobenzaldehyde in a mixture of 75 ml amyl alcohol and 25 ml concentrated H2SO4 and convert them into indole. Again please, some organisms utilize the amino acid and convert them into indole. This is a character of some organisms. They utilize amino acid and they convert them into indole. This indole is identified with indicator, means Kovacs reagent. Pure culture was inoculated in a peptone water and incubated for 2 to 5 days at 37 degrees centigrade temperature. Add 3 to 5 drops Kovacs reagent to 5 ml broth culture. Production of yellow color indicated indole negative. Yellow color production means this organism is indole negative. Next biochemical test performed was Methyl Red Test for re-identification purpose. Some organisms, they utilize glucose and they produce very large amount of the acid. When the acid is produced, the pH is decreased and comes to near 4.5. This pH is identified with the help of an indicator which is called as methyl red. So pure culture was inoculated in glucose phosphate peptone water and incubated for 2 to 5 days at 37 degrees centigrade temperature. Few drops of methyl red reagent were added and the production of yellow color indicated a negative reaction. So production of yellow color indicated a negative reaction. So Bacillus subtilis is methyl red negative, indole negative. And number 3rd biochemical test which was performed, that is named as catalase test. This test demonstrates the presence of catalase enzyme, an enzyme that catalyzes the release of oxygen from hydrogen peroxide. A drop of H2O2 3% that was placed on the glass slide and then a loop full culture of pure isolate was mixed. Air bubbles formation indicated the positive reaction. So Bacillus subtilis is catalase positive. Air bubble formation indicated the positive reaction. Next biochemical test performed for re-identification of this organism was Voges-Prosker test. This test describes the formation of acetyl methyl carbinol, which is also called as acetone, from dextrose. Acetone is oxidized by the Barrett's reagent to diacetyl, which produces a red color with gonidine residues in the media. Pure culture isolate was inoculated procedure. Pure culture isolate was inoculated into glucose phosphate peptone water and incubated overnight at 37 degrees centigrade. Few drops of Barrett's reagent were added mixed in solution containing potassium hydroxide 40 grams, creatinine 0.3 grams, and distilled water 100 ml. Development of pink color in 2 to 5 minutes indicated the presence of acetyl carbinol compounds. No change in color indicated negative results. No change in color indicated negative results. Next step was the examination of motility of Bacillus subtilis organism. For this purpose, hanging drop method was used to examine the motility of that particular organism. So first of all, a pure broth culture was selected. Then where a concavity was available in cavity slide, placed a small ring of petrol jelly, which is also called as Vaseline, around the depression. Then placed a flat cover slip on the bench, put a small drop or the broth on it, inverted the concavity slide on the cover glass so that the depression was exactly over the drop of the broth. Pressed down gently to provide an airtight seal, turned the slide over quickly and carefully. The hanging drop of culture should remain suspended in the center of the slide. Examined under the high dry objective in phase contrast microscope, reduced the illumination with the iris diaphragm and initiated examination from the side of the drop. Reduced the illumination with the iris diaphragm and initiated examination from the side of the drop. Distinguished Brownian motion by particles in the broth indicates the motility of the organism. Next step was the mass production of Bacillus subtilis. Pure isolate was spread out on the surface of the nutrient agar plate and was incubated for 18 hours at 37 degrees centigrade. खुला रखो. Pure isolate was spread out on the surface of the nutrient agar plate and was incubated for 18 hours at 37 degrees centigrade. For the confirmation of the growth, gram staining method was used. Then pure growth was inoculated in 4 rocks flasks containing nutrient agar. Then growth was harvested from rocks bottles by adding 20 sterile glass beads and 25 ml of normal saline per bottle. This is actually the process of mass production of Bacillus subtilis organism. Next step was the preparation of working spore suspensions. Next step was the preparation of working spore suspensions of Bacillus subtilis. Spore count, for this purpose, spore count in the suspension was determined by Breed's smear method, and spore count was 10 raised to power 12 per ml. Then from this stock suspension, working spore suspensions, they were made for further used. And when I say that working spore suspensions were prepared, were used, that were actually 10 raised to power 2, 10 raised to power 4, 10 raised to power 6 and 10 raised to power 8 per ml. They were made for further use. 10 raised to power 2 spore suspension was prepared by taking 0.0625 ml from stock suspension and adding in 100 ml of phosphate buffer saline. By doing this, we made 10 raised to power 2 per ml spore suspension of Bacillus subtilis for further use. And for making 10 raised to power 4 spore suspension, that was prepared by taking 0.125 ml from stock suspension and adding in 100 ml of phosphate buffer saline. By doing this, we prepared 10 raised to power 4 per ml spore suspensions of Bacillus subtilis for further use. And for making 10 raised to power 6 spore suspensions, that was prepared by taking 0.25 ml from stock suspension and adding in 100 ml of phosphate buffer saline. And for making 10 raised to power 8 per ml spore suspension, that was prepared by taking 0.5 ml from stock suspension and adding in 100 ml of phosphate buffer saline. Excuse me. So by adopting all these steps, we prepared the working spore suspensions. Means we prepared 10 raised to power 4, 10 raised to power 2, 10 raised to power 4, 10 raised to power 6 and 10 raised to power 8 per ml spore suspensions for further use. After making this all, after mass production of Bacillus subtilis, after preparation of working spore suspensions, next step was the medium preparation for STAF kit. Preparation of medium for STAF kit. This was the next step to be taken. So for this purpose, nutrient agar was prepared. Nutrient agar was prepared as a base medium with the following composition. Composition of nutrient agar is as under. Number 1, peptone 10 grams, beef extract 5 grams, agar 20 grams, sodium chloride 5 grams, distilled water 1000 ml, and 0.4% dextrose 4 grams per 1000 ml of nutrient agar. The above medium was sterilized and adjusted at the temperature of 48 degrees centigrade for further use. So this was the step for preparation of medium for STAF kit. This medium was sterilized and adjusted at the temperature of 45 degrees centigrade for further use. Collection of samples was the next step to proceed further. 25 samples, for this purpose 25 samples of each category were collected out of the survey conducted in district Faisalabad. Each category means milk of buffalo. 25 samples of milk of buffalo they were collected. 25 samples of urine of the goat they were collected. 25 samples of poultry liver they were collected, and 25 samples of poultry meat were collected out of the survey conducted in district Faisalabad. They were initially observed for the presence of antibiotic residues through application of STAF test. Number of positive and negative samples of each category they were collected and they are mentioned in table number 2. They are mentioned in table number 2, and I would like to share this table if it is possible. Table number 2. Inshallah, I will share it further. Each negative and 2 negative samples of each, 8 positive, 8 positive and 2 negative samples of each milk, urine, liver and meat, they were selected. Out of those 25 samples, 8 positive samples and 2 negative samples, they were selected. Then they were preserved further in the refrigerator for further use. We are discussing the preparation and optimization of ready to use kit conditions for swab test on animal food, and in this research we prepared and we optimized 3 things. Number 1, thickness of the media. Number 2, spore count of Bacillus subtilis. And number 3, incubation temperature. These 3 things were optimized, and the basic purpose of optimization was to get rapid results as early as possible in field conditions, so that animals having residues, they may be retained, their slaughtering may be delayed, and purpose of the research was that the feasibility of STAF kit under field condition, that should be noted, and to get more visible zones of inhibition under field conditions. So for this purpose we adopted all these steps. After collection of the samples, next step was the preparation of different medium thickness on plastic trays. Preparation of different medium thickness on plastic trays. In this step, 3 groups were made and named as group 1, group 2 and group 3, each containing 4 transparent plastic trays. Each containing 4 transparent plastic trays. Note it please. In this way, group 1 having 4 transparent plastic trays, A, B, C, 3, C and D. Group 2, E, F, G and H. And group 3, I, J, K and L. Each group having 4 transparent plastic trays. We are discussing the step of preparation of different medium thickness on plastic trays. For this purpose we made 3 groups having 4 transparent plastic trays. So in this step 3 groups were made and named as group 1, group 2 and group 3, each containing 4 transparent plastic trays. Each plastic tray was made up of polystyrene material. Each plastic tray was made up of polystyrene material having 13.5 centimeter length and 8.5 centimeter width. That plastic tray's length was Each plastic tray length was 13.5 centimeter and width was 8.5 centimeter, and depth of that tray was 1.5 centimeter. Group number 1 was containing 4 transparent plastic trays. They were named as A, B, C and D, and in each plastic tray in group 1, 2 millimeter thick layer of nutrient agar was made by pouring 20 ml of nutrient agar with the help of sterilized graduated cylinder. So in group 1 we have 4 transparent plastic trays. We used 20 ml of nutrient agar in each plastic tray to make 2 millimeter thickness of nutrient agar. Similarly, in group 2 was also containing 4 transparent plastic trays named as E, F, G and H. But here in this case, in each plastic tray 4 millimeter thick layer of nutrient agar was made, and to make 4 millimeter thick layer of nutrient agar we poured 40 ml of nutrient agar with the help of sterilized graduated cylinder in each tray. So by doing this, 4 millimeter thick layer was made in each plastic tray. And so on in group 3, which also contained 4 transparent plastic trays, and they were named as I, J, K and L. But here in this case, in each plastic tray, 6 millimeter thick layer of the nutrient agar was made by pouring 60 ml of nutrient agar in each plastic tray with the help of sterilized graduated cylinder. Then all the trays, after making 2 mm, 4 mm and 6 mm thickness in each 3 groups, all these trays, they were allowed to solidify. After solidification, all trays were capped on and sealed in sterilized double plastic bags separately. Each plastic tray was capped on and that was sealed, and each group was separately sterilized in double plastic bags. After preparation of different medium thickness, next step was the spreading of working spore suspensions, which we have made earlier. Working spore suspensions 10 raised to power 2, 10 raised to power 4, 10 raised to power 6 and 10 raised to power 8 working spore suspensions. Next step was actually the spreading of those working spore suspensions on the surface of nutrient agar of plastic trays of group 1, group 2 and group 3. We are actually optimizing the medium thickness, the working spore suspensions, as I have already discussed about this, that we had to optimize thickness of the media, spore count of Bacillus subtilis and incubation temperature. So for that purpose, step taken was the spreading of working spore suspensions on the surface of nutrient agar of plastic trays. Group 1 was containing 4 plastic trays A, B, C and D having 2 millimeter thick layer of nutrient agar. 10 raised to power 2 per ml spore suspension was taken and a sterilized cotton swab was soaked in 10 raised to power 2 per ml spore suspension and then was spreaded on plastic tray A having 2 mm thick layer of nutrient agar. After that, another sterilized cotton swab was soaked in 10 raised to power 4 per ml spore suspension and then spreaded on plastic tray B, which also have 2 mm thick layer of nutrient agar. After that, cotton swab was soaked in 10 raised to power 6 per ml working spore suspension already prepared, and then that was spreaded on plastic tray C having 2 mm thick layer of nutrient agar. And lastly, another sterilized cotton swab was soaked in 10 raised to power 8 per ml spore suspension and then agar spreaded on plastic tray D having 2 mm thick layer of the nutrient agar. After spreading this, group 2 was taken. This group 2 was also containing 4 transparent plastic trays which were named as E, F, G and H, and they were having 4 mm thick layer of the nutrient agar. And then working spore suspensions 10 raised to power 2 per ml, 10 raised to power 4 per ml, 10 raised to power 6 per ml and 10 raised to power 8 per ml spore suspensions were respectively spreaded over tray E, F, G and H by adopting the same method. Lastly, group 3 was taken, which was also containing 4 transparent plastic trays I, J, K and L. They were having 6 mm thick layer of the nutrient agar. Plastic trays I, J, K and L, they were spreaded with 10 raised to power 2 per ml, 10 raised to power 4 per ml, 10 raised to power 6 per ml and 10 raised to power 8 per ml spore suspensions respectively after soaking cotton swabs in each spore suspension separately by adopting the same method. Then all the trays, after spreading these working spore suspensions, all the trays of each group were sealed separately in sterilized double plastic bags separately to avoid evaporation. After that, next step was the placement of Neomycin discs and sample swabs. Next step was the placement of Neomycin discs and sample swabs. So after spreading of different spore suspensions, next step was the placement of this Neomycin disc of 5 micrograms on the one side of all plastic trays. Then next step was the cotton swabs. They were soaked in milk, in urine, in control negative samples, liver samples, in meat samples, and all these samples were taken in triplicate, 3 samples of each category respectively. After soaking cotton swabs, they were placed on the surface of nutrient agar of all trays of group 1 A, B, C, D, of group 2 E, F, G, H and of group 3 I, J, K and L. After soaking cotton swabs, they were placed on the surface of nutrient agar of all trays of group 1, 2 and 3, trays having different medium thickness 2, 4 and 6 mm and also different spore count 10 raised to power 2, 4, 6 and 8 per ml in the plastic trays of all groups 1, 2 and 3. Then trays were capped on and sealed in double plastic bags. So placement of neomycin disc and placement of all these samples on the plastic trays in triplicate was done. After that, next step was the incubation at different temperatures. Incubation at different temperatures. Group 1 and group 2 and group 3 trays, they were sealed separately in different plastic bags. They were incubated at 35 plus minus 2 degrees centigrade temperature. Group 1, 2 and 3 trays were incubated at 30 plus minus 2 degrees centigrade temperature. And group 1, 2 and 3 trays were sealed separately in double plastic bags, incubated at 25 plus minus 2 degrees centigrade. So note it please, we have incubated all trays of group 1, group 2 and group 3 at three different incubation temperatures. Number 1, we incubated group 1, 2 and 3 trays at 35 plus minus 2 degrees centigrade temperature. Number 2, we incubated group 1, 2 and 3 trays at 30 plus minus 2 degrees centigrade temperature. And number 3, we incubated group 1, 2 and 3 trays at 25 plus minus 2 degrees centigrade temperature separately. And then results were noted after 6 hours initially. Again, results were noted after 6 hours onward after every 1 hour. And so on up to 24 hours. We noted the results initially after 6 hours, and after 6 hours, after every 1 hour we noted the results up to 24 hours, and results were shown in appendix 1, 2, 3 and 4. Soaking. Sterilized cotton swab soaked in poultry and meat sample are shown in this picture. Note it please, like this. This is poultry sample soaked, and this is liver sample. Cotton swab soaked in poultry liver sample. Cutting of sterilized cotton swab soaked in poultry meat and samples, like this. And this is the placement of swab soaked in liver sample, placement of these swabs on plastic trays. So after that, results so far as the results are concerned, we are going to discuss these results with you to avoid the wastage of time. Results of milk, urine, liver and meat samples. After adopting all these procedures, summary. After adopting all these procedures, results were noted initially after 6 hours, as I have mentioned earlier, and then after 6 hours we noted the results after every 1 hour and 24 hours. From results it became clear that maximum zone of inhibition around each category of sample swab was obtained at 4 millimeter thick layer of nutrient agar, and maximum zone of inhibition obtained at 10 raised to power 4 per ml spore suspension of Bacillus subtilis, and rapid results were obtained at 35 plus minus 2 degrees centigrade temperature of incubation, and these results were obtained within 8 to 9 hours. So we optimized that at 4 mm thick layer of nutrient agar, on which 10 raised to power 4 per ml spore suspension was spreaded, and which was incubated at 35 plus minus 2 degrees centigrade temperature, was incubated at this temperature. Within 8 to 9 hours, maximum zone of inhibition was noted. So finally, cost per sample was also calculated, and it was found that 3.33 rupees per sample costs only. So this field kit is very cheap, not very costly. I mean, this is good for field conditions, so we have to adopt this. Calculation of the cost per sample was done in detail. We will discuss it later on, inshallah. So this was all about my research work done. This kit may be used in slaughterhouses to check the antibiotic residues in the animals. If within 8 to 9 hours samples are found positive, then we may retain the animals and we may delay the slaughtering of those animals, so that consumers may not suffer from antibiotic resistance problem. So this kit is very effective in slaughterhouses, in poultry farms, in field. Kit is not very expensive, so please use it. This was all about my research. Thank you very much.